The overall goal of this procedure is to isolate leukocytes from the human Decidua Basalis and Decidua Parietalis. This is accomplished by first dissecting the human decidual tissues from the placenta. Next, the decidual tissue is dissociated by mechanical disaggregation and enzymatic digestion. Then, the cell suspension is filtered using a 100 micrometer cell strainer, washed with 1x PBS, and centrifuged at 300 G for 5 minutes at 4 degrees Celsius to obtain a cell pellet. Finally, the cell suspension is loaded on top of the density gradient solution and centrifuged, and the interface containing leukocytes is collected. Ultimately, isolated leukocytes can be used for immunophenotyping, cell culture, and functional studies.
The main advantage of this technique over existing methods, such as mechanical dissociation or enzymatic digestion, is that it combines mechanical tissue disaggregation with an automatic tissue dissociator, enzymatic digestion with acutase, and leukocyte isolation using a fecal gradient. This protocol has been proven to preserve cell surface antigens and cell viability, and its implications extend toward the understanding of the functional and phenotypic properties of infiltrating leukocytes at the maternal-fetal interface related to the pathogenesis of perinatal complications.
The collection and utilization of human samples for research purposes were approved by the Institutional Review Boards of the Eunice Kennedy Shriver National Institute of Child Health and Human Development, National Institutes of Health, Department